All right, next up we're going to talk about what happens outside of the mitochondria. So we're going to break down glycolysis just a little bit more, and then we'll go into the process of fermentation. Glycolysis occurs in the cytoplasm, but outside of the mitochondria. It consists of a series of 10 reactions, each with its own enzyme. Now there's going to be a first step, which will be the energy investment step. At this point, 2 ATP are going to be used to activate the glucose. Once they are activated, that glucose is going to split into 2 G3P molecules. After that, we start harnessing energy. And the energy harvesting step, this is where we have the oxidation of the G3P occurs by removing the electrons in the hydrogen atoms, or hydrogen ions. 2 electrons and 1 hydrogen ion are going to be accepted by NADH, or NAD+, which will result into NADH. 4 ATP are going to be produced by substrate level phosphorylation. In other words, at the enzyme level, we're going to be producing ATP. An enzyme passes a really high energy phosphate to ADP, making it ATP. This stage, there's going to be a net gain of 2 ATP. The reason why we call it a net gain is because there are 4 ATP produced, but we had to invest 2 ATP to get the ball rolling on this one. At this point, both G3Ps are going to be converted to pyruvates.
Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of 2 distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, 2 ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the 2 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell, and NADH. During the initial step of glycolysis, energy from ATP is invested into the process. 2 phosphates from ATP are transferred to a glucose molecule. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. During the energy harvesting steps of glycolysis, each 3-carbon molecule is converted through a series of steps to pyruvate. Electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is produced. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
So that's going to be our overview of glycolysis. Here we have a diagram that kind of shows everything, shows all of the steps. The biggest thing I want you to know is about this energy investment step. 2 ATP go in, 4 ATP come out, thus we have a net gain of 2 ATP. Glucose goes in, pyruvate comes out. That's the ultimate goal of what's happening during glycolysis. So that's going to, yes, so that is our glycolysis. There are, I'm sorry, some of my slides are a little bit out of order. One moment. So complete glucose breakdown requires an input of oxygen in order to keep that electron transport chain working. But there are some cells that have to continue working, continue producing ATP even when oxygen is scarce. So how are they going to do that if oxygen is limited? Very good, through the process of fermentation. Fermentation is an anaerobic process, meaning that it can occur in the absence of oxygen. In animal, this process can produce a very limited amount of ATP in the absence of oxygen. In animal cells, pyruvate is the end product of glycolysis. It is reduced by NADH to lactate. Anaerobic bacteria can also break down pyruvate to form lactate, or they can break it down to form carbon dioxide and alcohol. It just depends on the organisms. Great example of this would be yeast. Yeast are an excellent example of organisms that generate ethyl alcohol and carbon dioxide as a result of fermentation. If oxygen is available to the cell, then the pyruvate is going to enter the mitochondria for aerobic respiration. Here's an overview of fermentation. If you notice, the steps from here to here should look an awful lot like glycolysis, and that's because it is. The very first step the first part of fermentation is just the production of pyruvate. That pyruvate can then break down into lactate or alcohol, whether we're looking at, depending on whether we are looking at plants or animals or the specific type of organism. But you'll notice that the only step in which we actually produce ATP happens way back up here in glycolysis. So the process of fermentation yields only 2 ATP. 2 ATP is significantly less than the 36 or the 38 ATP that we could have gotten if we had undergone the process of cellular respiration. So why would we do this? Why undergo fermentation? Well, fermentation has its own advantages and disadvantages, but there are a lot of organisms that will undergo the process of fermentation, and there's a huge variety of foods that we consume which are actually byproducts of fermentation by microorganisms. Foods that are produced by fermentation tend to last longer because the fermenting organisms have removed many of the nutrients that would normally attract other organisms. In other words, by breaking down all of the glucose into pyruvate and alcohol and carbon dioxide, many of the nutrients are gone, so very few organisms are going to be attracted to that particular food source. Here's a great example of some foods produced by yeast fermentation. Bread is produced by the fermentation of yeast. The rising of the dough is dependent on the carbon dioxide produced by the fermentation of yeast, and the ethyl alcohol produced by the yeast evaporates during the baking process. Ethyl alcohol in beers and wines are produced when yeasts ferment carbohydrates. The fermentation of fruit yields wine. The fermentation of grain yields beer. Yogurt, sour cream, and cheese are produced through the action of various lactic acid bacteria that cause milk to sour. Milk contains lactose, which these bacteria can then use as a carbohydrate source for fermentation. So for example, yogurt is made by adding lactic acid bacteria to milk and then incubating it to encourage the bacteria to convert the lactose. So a lot of organisms do this. Why do they do it? Obviously, they're not doing it for our sake, but they are doing it because there are many advantages. One of the advantages of fermentation, it provides a very quick burst of ATP for muscular activity. When you're working out, when you're out of breath, sometimes you just can't breathe enough to keep your muscles going. So your body can undergo fermentation to get that quick burst of energy that is needed to keep you going until you're able to catch your breath. When muscles are working vigorously for a very short period of time, that lactic acid fermentation provides ATP. There are some disadvantages to fermentation, though. Lactate and alcohol are toxic to cells. Lactate changes pH. It causes your muscles to fatigue. This is due to an oxygen debt. If you work out too much, and you're not able to breathe or obtain oxygen as quickly as your body is using it for cellular respiration, you'll start to undergo fermentation. That lactate can change the pH, and it can wear out your muscle fibers. Yeast can actually die from the alcohol that they produce during fermentation. And fermentation's not all that efficient. 2 ATP are produced per glucose molecule during fermentation. It's equivalent to 14.6 kilocalories. Complete oxidation of the glucose could yield 686 kilocalories. So it's a very inefficient process. Only 2 ATP per glucose are produced compared to 36 or 38 ATP molecules produced by the cellular respiration. So the efficiency is only about 2% of the total possible energy that could be obtained from glucose breakdown. So here's just kind of a overview of fermentation. The input is glucose and 2 ADP molecules. The output is 2 lactate or 2 alcohol and carbon dioxide molecules plus 2 ATP. So it's a very sort of small net gain from fermentation. All right, in this chapter we talked about cellular respiration, how it is an aerobic process. We talked about how glucose is broken down into water and carbon dioxide. The process of cellular respiration contains 4 phases: glycolysis, the prep reaction, citric acid cycle, and electron transport chain. Then we went into fermentation. Fermentation involves glycolysis followed by the reduction of pyruvate by NADH to either lactate or alcohol and carbon dioxide, depending on the organism. This is only going to result in 2 ATP molecules, and the products of fermentation are often used in baking and in brewing. Oh, and advantage is that it does provide a quick temporary burst of ATP. And that's it for this chapter. Please let me know if you have any questions or comments.